Okay, we better keep moving because I've got a bit to cover. So I thought I might just provide you with an overview of what we were speaking about outside, because unfortunately most of the biology is invisible. So the only way you can really satisfy yourself that it's actually there is that your stubbles or your organic material is decomposing, but you can't readily see it. So you need to use very high-powered microscopes to look at it, or otherwise fancy DNA-based approaches to identify what's present. So we use mostly DNA-based approaches now, and it's because microbes are living things. They all have DNA, and we can identify using markers what's present in the soil. So there's a whole lot of things present in the soil, going from small, the bacteria and the archaea. Oh, that's cool. It's like a spotlight. That's nice. Then the fungi, we've got the fungi, the second row down, and finally along the bottom we've got the macrofauna, and they're the things that we're more likely to see. So we were able to see some earthworms, the rosy tip earthworms, out in the paddock before. So they all work together. They work as a community called the food web, and so an impact on one will have a massive impact on the others. In terms of the impacts on the plant, we're probably more interested in the bacteria and archaea here, because they are in fairly close proximity to the plant, but they also have they're also the chemical engineers of the system. They're the chemical factories, so they process nutrients from plants and from fertilisers and other inputs and make nitrogen available. So we measure them by their numbers, who they are, and we also look at the environment they're living in to get a sense of what factors in their environment tend to encourage their presence and abundance and activity. The other thing I wanted to just say is that microbes are everywhere. Now, if we watch the telly, we'll hear about microbiomes and the gut microbiome. We hear all about those. Chris Hemsworth even talks about microbiomes. But there are plenty of agricultural microbiomes that are way more important because they have global impacts. But for most significant microbiomes for plant performance, they are derived from the soil. So they start off in the soil, they're found on the stem of plants, they're found on the leaves, and they're also found inside the plant. They're called endophytes. And so therefore, the point really is that microbes in the soil are a repository, a very valuable repository of the microflora. So this was done quite a while ago by a colleague of mine, Laurie Phillips, and she extracted the DNA from a vertisol, a heavy soil from this area. I think it must be Scrime because we did a lot of sampling there. And this basically shows that if you do DNA sequencing and you match your sequences with libraries, the global libraries of sequences, you can identify the types of organisms that are present. And you can tell straight away from this list that there's a lot of different things present in our soil. And we're going to talk about one group in a little bit more detail, and the group we're going to talk about are the nitrifying bacteria. They're those things that mineralise ammonium to nitrate and nitrite that the plant can then use. So they're very important. And yeah, the point of that little schematic over there is to show that microbes work together, and this is best illustrated by the nitrogen cycle, which shows a whole lot of different types of species or genera or whatever groups that depend on each other to convert nitrogen into a form that they can then take and use. So it's a bit of a bucket brigade thing that's going on. So interaction is very important. So another important point to make is that we can measure microbes at scale. There's a lot of work happening at the scale of geography, continental scale. There's a program that has happened over the past few years that GRDC supported that mapped the biology of the Australian soils, if you like. And so we now know that where things are occurring and in what levels of abundance. And there's a lot of things that occur in southeastern Australia. So at the continental scale, we know that latitude is really important in the types of communities that we have. We also know at the regional scale, we can ask the question, well, what happens with land use? Does land use affect the microbial communities? Well, yes, it does. We've been able to show in studies that the way you manage your regional soils will have an impact on the types of communities that are present. At the local scale, the paddock scale, which is probably the most relevant to all of us, we can assess how inputs into our systems impact on the microbial composition of our communities. And we can ask some really important questions about, tillage and rotation and pesticide application and things like that. So they're very important, but so is the context of continent. So we need all three scales basically, because we need to know what groups are going to occur in this region and do the work at the paddock scale. So we need to have a bit of a wider context. The other thing I just wanted to mention, and couldn't do it outside, is the importance of pore structure in the soil. It's important that we have a range of different pore sizes in soils, because different components of the microbial community or the biological community have preferences for different pore sizes. So the larger organisms, the macrofauna, tend to habitate the larger pores, and the smallest ones, the smaller pores, tend to be home to bacteria and some fine fungi that can grow into those smaller pores. So it is important that we maintain pore structure so that we maintain a healthy home for our microbial community. The only point, I noticed the title was all about microbes and soil structure. The point I want to make is that in a gram of soil, you've got about 1,500 kilometres of hyphae. That's what it looks like if you look at it under a microscope. And so it's pretty, if you can believe that, then you can understand at the lower magnification, you can see the impact it has on binding soil crumbs together. And it's a critical role of the microbial communities in our soil. It acts like a string bag, and it can just hold together all the crumbs that create that very important aggregate architecture that we want. And that then aids water infiltration and also oxygen exchange or gas exchange, which is also important. Microbes also produce substances by their action. There's a substance, that greeny fluorescence is a product called glomalin. It's produced by vesicular arbuscular mycorrhizae, but it's like a glue. So it can glue together some of the smaller particles of soil. And that works together with bacterial polysaccharides, which you see that sticky substance down there that's holding the... where's my spotlight? It's holding the cells together, and it's actually working towards binding the particles together as well. It's actually okay, oh boy. This is just the overview. Okay, I'm going to skip. Okay, so an important point to know is that microbes are in the soil and they just want to grow. and they will take their energy from whatever they can. So this is a result of a DNA analysis which enables us to identify what are the important functions that the whole of the community are performing. So these functions summarise, I guess, what the main functions are that happen, and they're all related to growing. If you look in other and blow that up, others 27%, there's a whole lot of other things that go on related to nitrogen metabolism, potassium, sulphur metabolism, dormancy, and they're important things in agricultural systems. But if you take a slice of that 27%, only less than 5% is devoted to nitrogen activity. So in the scheme of things, the microbial community as a whole devotes a fairly small amount of its gig to nitrogen metabolism. And of that nitrogen metabolism, it's all about ammonia assimilation. So it's about growth, essentially. So this is the main game. I was going to talk to you about microbial nitrogen properties, and I'll just skim through this. So the take-homes really are that the tactical use of inputs should factor in microbial turnover of nitrogen. So they should factor in that whole microbial turnover story. So we need to consider immobilisation, nitrification, mineralisation in both space and time. And there's some work that's been done around the place that tries to explain how the microbial turnover aspects of the nitrogen cycle work. The other take-home message is that estimates of nitrogen supply to the plant really should take in the microbial capacity to transfer nitrogen. I think in doing so, we will develop much better regional measures of what nitrogen inputs are required. So there's just a few points here. The first one, I can't emphasise it enough, and that is that the soil nitrogen cycle relies mostly on microbes. So it's important that we consider the microbes in those cycles. If there's all plant available nitrogen has been processed by the microbial community in your soil. And every region has its own fingerprint, really, of the microbes that are involved in that process. So that fingerprint must be taken into account in this area. And I think I mentioned about growth being the main game. And so carbon and nitrogen are both very important energy elements for microbial communities. So just looking at the nitrogen element, we know that it's essential for growth for both plants and microbes are better than plants are at competing for the nitrogen, better than plants yet. So we know it's a balancing act. We know that there's a tension between supply of nitrogen for plants and demand by, I'll talk in deficiency and surplus, I think it's better to say. So there is a balance between deficiency and surplus in a microbial community and in a plant community. There are different forms of nitrogen, and there are about 9 forms, I think, and they're all in different oxidative states. So some of them are very oxidised, so nitrous oxide, nitric oxide, and some are very reduced, like ammonium, for example. It has hydrogen attached to it. And that is due to the work of a whole lot of different groups of microbes that supply these different forms of nitrogen. And I don't know, you've probably heard some of these names of processes before. There's nitrogen fixation by microbes associated with legumes, depolymerisation, so taking the big molecules from the, the plant residues and forming smaller molecules. There's immobilisation that takes mineral nitrogen and ties it up into the microbial community or into the plant. Mineralisation, which then oxidises the nitrogen and makes it again available to plants. Nitrification and denitrification. So there's a whole lot of microbial processes that we need to take into account if we want to work out what's going to be available for the plant. So Rob Norton did some work which I thought was really neat, which used partial nitrogen balance equation on regional soil. So he surveyed a number of farms in the region and worked out importantly that, so I should explain it a bit. So he looked at these regions here and he worked out the partial nitrogen balance here, and he then assessed whether these figures, the partial nitrogen balance was less than 1, in which case more nutrient was added than removed, or it was greater than 1, more nutrient removed than added. In the case of greater than 1, because all of those regions are greater than 1, there was a depletion and drawdown of soil reserves. So in current fertiliser systems, there is over the last 3 years, there has been a depletion and drawdown of soil organic nitrogen reserves. And this is illustrated here. If you just look at these graphs here, you can see that for the Mele, 60% of the farms had partial nitrogen balances greater than 1, so there was drawdown, and in the Wimmera it's about 50%. So there's drawing down of these of nitrogen in these soils that I guess we need to really address. So to address that, we really need to understand microbial nitrogen drawdown. And to do that, we need to consider, I guess, how the microbial community impacts on, or how it impacts on crop residue breakdown. And you've probably all heard about nitrogen tieup by John Kierkegaard and a whole range of other people. And basically, what happens is that when you add a crop residue, it has plenty of carbon in it. And so what it does, it tends to increase the microbial biomass in the soil because the microbial biomass uses the carbon as an energy source, and then it can decompose that crop residue. However, in using the carbon to grow and to decompose, it also has a nitrogen requirement. And as I mentioned before, it is better able to draw nitrogen from wherever reserves they can grab it than, say, the plant is. So the plant gets the rest of the nitrogen after the microbial demand for nitrogen is met. And so that's basically what this tells us. And so what this shows is when you add a crop residue, this biomass combined with that biomass, which is also in there, will grow and it will immobilise nitrogen, so it pulls it out, it needs nitrogen, it'll take it out, and then it dies, and then it releases it, so it mineralises it. And all in the meantime, the plant sits there waiting for its own source of nitrogen. And you can... So there's also, these fertilisers. And crop residues have C to N ratios. So I guess I want to skip a few, that's all. So because there's different C to N ratios, the higher the carbon, or the higher the ratio, the more carbon, the less nitrogen, you will see more tie-up of nitrogen. So the lower the carbon to nitrogen ratio, so the more nitrogen, there's less demand for growth, and so there'll be less nitrogen tie-up. And so with a few rules that were created originally by Fran Hoyle and Dan Murphy, there's a few assumptions that have been made, and you can basically model it and show that with increasing C to N ratios, so down one end you've got, say, field P, which has a ratio 32 to 1, so a fairly low ratio, and wheat has a ratio of 80 to 1. And if you run those assumptions in a model, you will show that you will get, so this is kilograms of nitrogen, and the different lines are how much residue you put in to start with, the different tonnages. But you'll see basically the same thing in the model. The greater the C to N ratio, the greater the deficit of nitrogen in the system. So with that deficit, you will be, the crop and the microbes will have to look elsewhere for their nitrogen to make up for their demand for nitrogen. It's a complicated story. Gupta made the observation, I think we've all understood, that the bigger the microbial biomass, the bigger demand for nitrogen, the bigger demand for nitrogen, so the greater it will take up. It'll take up more nitrogen, but it also has a potential, when it dies, to release more nitrogen. So the more you grow the biomass, the more you will increase its immobilisation of nitrogen, as well as the later release of nitrogen. And if you can get if you can time the growth, the turnover well, you can manipulate when the nitrogen is going to be released back into the system. I'm going to skip a few because we haven't got time. I just want to say a couple of things about nitrification, which is a really special part of the biomass. So it's part of the big biomass, but the microbes in that part of the biomass have a specific function related to the nitrification, or the conversion of ammonium to nitrate, which then the plant can use. And there are products around which inhibit that from happening, and they're not always very good. They're a little bit variable and a bit hit and miss. The candidates that are associated with that, they belong to the bacteria, and here in this donut structure are all the bacteria you'll find in your soil. But the nitrifiers are present in this big abundant section, the Proteobacteria, and they're also present in Crenarchaeota. So the nitrification is carried out by bacteria in the Proteobacterial group, and they're also carried out by archaea, which is another group. So it's a fairly diversely held function, which is a good thing, because because you can be adapted to the conditions. So who is dominant in the Birchip area, in the Birchip region? If you look in the Proteobacteria, so nitrifying bacteria, in a heavy soil and a light soil, there are three main families, with the blue, the yellow, the family Bradyrhizobiaceae, and this other guy whose name I can't really pronounce very well. And those families are present in all three places. This is fairly brand new. And if you drill down into those families and look in the genus levels, the main, or the things that we find are Nitrosovibrio, Balnemonas, and thioalkylo vibrio, which I can just say. And this is all discovery. No one has ever found this before. What's interesting is that if you open up a textbook and they talk about nitrifying potential of soils and how we're going to develop inhibitors for particular groups, they talk about nitrous amonis and nitrobacter. And guess what? We can't find them in our soils. So we're applying targets or target chemicals to things that we don't have in our soils. So wouldn't it be better to develop approaches when we know what's present in the soil? Oh, I should think we do. It would be better. And this is another group. We talked about the Proteobacteria, another group here on a heavy and a light soil. So this is another group of nitrifying bacteria. And you can see that there's, 4 different types of genera that are present in a light and a heavy soil. And interestingly, in the light soil that dry out fairly quickly at different depths, you can see that, say, follow the grey, which is this unclassified, we don't even know what he is. He tends to be in 10 to 20 centimetres, so 10 to 20, 10 to 20, 10 to 20. So some of these nitrifying species have adaptations to moisture conditions. So we need to know a little bit about the biology. I'm going to stop right now. Take home's just repeated, and I would just want to thank all these people who contributed to the presentation. So thanks.
Pauline, at the moment there seems to be a lot of people pushing for regenerative farming, regeneration farming. No chemicals, no fertilisers. They're saying they're lifting the organic matter from 1.5 up to 5 and 6 in 5 or 6 years, they say.
Okay.
Massive. I don't know how they do it, but that's what they're saying.
I don't know either.
And but there seems a big push at the moment. The CMA are pushing it. There's a lot of farmers, a lot of field days on at the moment around that issue. What is that? I'm asking, is that possible, do you think, to increase the soil by that much organic matter? And if we did that, what effect would that have on their nutrient levels, nitrogen, moisture potential, that sort of stuff?
Yeah. Is it possible? It depends on where you are and what your starting point is. But in each region, and I think each region will have its maximum achievable limit for soil carbon, and that's because it's a combination of the use of the carbon, the tie-up of the carbon in the soil as humus, which is the major fraction, or whether it's driven off as CO2 by the microbes. So there's some kind of a dynamic there that in some climates, particularly, so for example, in a climate where it's you have high levels of high temperatures and moistures, you will get more CO2 driven off, particularly where you've got carbon on the soil surface, say compared to a place where the temperatures are lower. So I think each region, based on its climate, will have a different capacity, a different holding capacity of carbon. So that's one thing. But it depends on the starting point too. It depends on where you are. So there is a possibility, if you start low, to be able to build it fairly significantly. But I find it difficult to believe that you would be able to build it that much. Maybe it's kind of selective reporting of data. So I would be asking, where is this happening? What's the climate like? What was the starting point like? What did you measure? What we talk about carbon, but what forms of carbon? There are zillions of forms of carbon. If you measure total carbon, it doesn't budge. You can measure it, it incrementally changes, but if you're measuring the dynamic component, particular organic matter, which is the the component that microbes use and turn over really quickly, that will go like this. So it depends on the fraction that you look at, as well as the overarching climatic conditions. So,
What's the level we get it to, say, Birchip, you think?
so what's this?
Realistic. I mean, it's about one or something like that. Two?
Oh, are you going to quote me on this?
No, what's a realistic level that we get?
Two, maybe? Two, I think it would, yeah, like if you add pasture in rotation or something that's going to retain a lot of carbon, you might be able to do this. What's the highest carbon levels reported in this room? You know, what's the maximum achievable? You know, what has been reported in the region? Is it 2%? Well, that might be an upper limit. Okay, is it 1.5%? It might be half a percentage. Yeah. It's a difficult question, but yeah. But I think this whole thing about regenerative agriculture, I think 20 years ago it was called conservation tillage or conservation agriculture, and now we're calling it, and then it used to be sustainable agriculture, and now it's regenerative agriculture. So as a term, I don't have a problem with it. It's the claims that kind of go with it that I think is, yeah.
We've got time for one more question. Is there one out there? Yep.
Thanks. I'm told that those microbes are killed by artificial fertilisers. Or they're either killed or they go underground because it's like...
Yeah. Yeah, well that I think they're...
The way they break down the stubble
yeah.
is really critical.
Yep.
Really, really important for farmers.
There has been, my colleague who worked at Longrenon at the Scrime site, Roger Armstrong's Scrime site, showed that if you continued adding fertiliser to a stubble retained system, what you did was you suppressed the breakdown of organic nitrogen, because it's a different set of microbes that do that role compared to those that convert fertiliser nitrogen. So as you added fertiliser nitrogen, you tended to suppress the breakdown of nitrogen from organic material. That was at one site, and so that probably needs to be validated. But yeah, I guess there is whenever you add fertiliser, you will have an impact on other components in that massive nitrogen cycle. It's inevitable. We're yet to understand it, except that nitrification or mineralisation of organic nitrogen and nitrogen fixation can be suppressed by fertilisers for different reasons. Yeah.
Sorry to cut this interesting discussion off. I'd just like to say thank you again to Pauline and Mel. We've got gifts for them, but I left them out at the saw pit, so I'll give it to them at lunchtime. Are you hanging around later on if people have got more questions? Yep, Pauline and Mel will both be here all day. If you've got more questions, go and see them. And other than that, it's time to move on to the next station. So follow Amy with the red flag, and she'll take you to the adjuvants demonstration on the other side.